Okay. Well, thanks, Doug, and thanks for inviting me out here. And I've had a great time visiting with people and visiting your campus throughout the day. I'll be telling you about work we've been doing at near-infrared quantum ghost imaging of living and unperturbed plants. So first, before I get into quantum imaging, I just want to talk about the system and why plants are important. So plants can take, sunlight, water, and dirt, and with that as starting materials, can convert that into very useful things. And these are things, plants, we can eat them. They can provide materials and fabrics that we wear and use. They're the basis of a lot of pharmaceuticals and a lot of drugs. You can also convert them into fuels that we can burn in our cars. So plants can do this, can take carbon dioxide and make higher element carbon things out of them that are very useful for us as a society. And as you may know, that plants do this through a process called photosynthesis. You may remember this from your high school days or undergrad days, because I think most of you are physicists. So I'm not going to spend too much time talking about photosynthesis other than to say it's a complicated problem. You have many different proteins which are in there for light harvesting, for carbon fixation, for electron transport, for building these larger molecules in the Calvin cycle. I just want to say it also uses specialized cells and specialized cell types. It's a very complicated biochemical and biological process. And I think one of the best ways we have of looking at complicated biological and biochemical processes is microscopy and imaging. And I think microscopy and imaging is very important because you get to see the molecular players in their real physical context and how they interact with one another spatially and temporally. You could think of sort of a pedagogical example, but think of a cell as a collection of Lego blocks. Some of these are RNA, let's say the green are RNA, the red are DNA, the yellow are lipids, some of these are proteins. We have a lot of ways in biochemistry of just counting how many of that we have in a cell, what I like to call grind and find kind of chemistry. But you can get information from what's in a cell, but you get a lot more information, I think, when you see these things assembled into a functioning unit. So these Legos assembled into a Lego engine, you automatically now have an idea what the function of this is. What's better than I think a static picture of an engine is actually a movie. A 3D movie of an engine gives you even more information. This is just a movie off of YouTube of a 4-stroke, 4-cycle engine. You have your 4 strokes are suck, squeeze, bang, and blow. But you get to see how those 4 cycles go from sucking, squeezing, banging, and blowing. You get to see how chemical work in terms of this explosion is turned into mechanical work in terms of the motion of the piston. You get to see how that linear motion of the piston is turned into a rotary motion by the crankshaft. And you get all this type of information from a movie of this process in 3D that you just do not get otherwise. I think it gives you a lot of physical intuition onto what's going on in a system, is to actually take pictures of it, take movies of it, see how things change over time. So, that being said, I think microscopy is a very powerful tool to understand biological systems. And, really a picture is worth 1,000 words. You get a lot from pictures. But, the way we typically do fluorescence imaging or fluorescence microscopy, or the way I've been doing optical microscopy, we shine lasers into these things. And, plants, plants respond to light. We all know plants, things like sunflowers undergo heliotropism following the sun. But also they're in photosynthesis, there are light dependent and light independent processes in photosynthesis. And the reality is anytime you shine a light on a plant, you're changing its physical state. You're changing it to something else other than it is in a dark or resting state. So that's one key hurdle, is plants respond to light. The other key challenge you face with doing microscopy of plants is sort of just general to any type of fluorescence microscopy or imaging, is that we're often labeling these molecules with things like fluorescent proteins or antibodies or dyes, and you try to make sure that label doesn't perturb the dynamics of the protein you're studying. But you have the worry anytime you label something with a fluorescent indicator that you're perturbing its dynamics. So, microscopy and imaging, I think, are very powerful techniques to study complex biological systems. But when you go into plants, you face these 2 key challenges. they respond to light, and the labels can perturb the dynamics you want to study. And so I'm going to tell you how with quantum methods we can kill two birds with one stone. We can basically use quantum methods to get rid of the need for any type of label. We can go label-free imaging. We can also get rid of this need for a high laser fluence, which could potentially damage or perturb the system we want to study. So I'm going to tell you how we do this, how we get rid of those two hurdles. We're doing a technique. I'm going to mainly be talking about a technique called quantum ghost imaging that was developed 30 years ago. And in this technique, you take a photon at high energy from a laser, you split that one photon into two photons at lower energy. These are typically called your signal and idler photons. Your signal photon in our scheme just goes through free space, and he's relay imaged onto an imaging sensor, something like a camera. Your idler photon, it goes through a target you're interested in imaging, and it's relay imaged onto a single element bucket detector. So this detector, there is no spatial information whatsoever from this. It's just, did we see that photon or not? And what's interesting about ghost imaging is if you correlate photon arrivals on this bucket detector with photon arrivals on this imaging sensor, from that combined information, you can build an image of this object, even though the light hitting your imaging sensor never physically probed or went through your object. And so I don't know about you, but the first time I saw this, I was like, how in the hell could this possibly work? How could you build an image using photons which never went through your sample? And but I think the easiest way to understand how this works is just conservation of photon momentum. That when you have a pump laser with well-defined momentum of, say, h bar kp going this way, very clearly left and right, if one of these two photons goes up a little bit, like it's exaggerated in this diagram, the other photon has to go down a little bit because the vectorially h bar k of the p of the pump and h bar k of the signal and h bar k of the idler have to sum together. So when you measure this guy on this imaging sensor, you measured his direction through free space, you measured his k vector. Now you know for a k vector here, there's a matched k vector position down here. And if you see a photon through here, you know for every location here whether the photon made it through or not, or how often it made it through. And that, I think, is the easiest way to understand ghost imaging is in terms of conservation of photon momentum. You can think of this guy as giving you spatial information, this guy giving you his transmission information, and you get a full image by the correlation of the two. Now, I think an analogy I like to make is quantum ghost imaging. It's like a game of Battleship with quantum entangled light. You have your imaging sensor, which says, hey, I saw a photon at coordinate G4. Then you go ask your bucket detector, did you see that photon? If it did see it, is in this picture, it's a hit. And so now for this coordinate position in that path, you're not really even imaging in. The photon made it through, so your sample is fairly transmissive at that coordinate. And you build up grid point by grid point by grid point, a map of this transmission in that sample. So I think ghost imaging is a really cool way to image stuff. You can image objects with light which never really went through your sample. But in addition to being really cool, I think it's actually really useful for certain things. And the two key strengths I see of ghost imaging are this. The first is these two photons, or signal and idler, they don't have to be degenerate in energy. So you can be probing your sample at one wavelength and looking at its transmission with the bucket detector, and you can be doing your imaging in, say, the visible portion of the spectrum, where we have much better camera technologies and imaging technologies. So this allows you to probe at one wavelength, say in the infrared, where things like vibrational absorption give us contrast, and do your imaging in the visible. So this is one key strength, is this sort of wavelength shifting. The other key strength, as I see it, especially for ghost imaging, is that when you generate an entangled photon pair through spontaneous parametric down conversion, those two photons are generated simultaneously. I mean within hundreds of femtoseconds of each other. And because of that, if you can time photon arrivals very precisely, and you're looking for coordinated photon hits on these two detectors, you can beat down a lot of other sources of noise because you're looking for this coincidence. And that includes detector dark counts, noise, stray light. what you're looking for are two photons arriving nearly simultaneously. So it allows you to beat down a lot of other sources of noise you typically have in imaging experiments. And you can do this type of imaging at a very low light flux. We're doing imaging of plants, I'll show you later, with a photon flux 50 times less than a moon-filled night. So the plants, when we're looking at them, basically think it's night. So, so I told you about the two key strengths of quantum imaging. I'm going to say these map really well onto these two problems. This wavelength shifting is how we can get rid of labels, because we can do probing in the infrared where vibrational absorption gives us contrast. And the fact we're doing this coincidence detection allows us to get rid of the high light fluence that can be perturbative to the plant. And so I would say, if you're disturbed about this AI graphic being a little bit too graphic here with killing these birds, I'm just doing what I can to remind you that Schrodinger thought about using quantum methods to kill cats. I'm just talking about using it to kill birds. So keep that in mind. So if you talk about what's in a plant, and you're a physicist, well, we could just assume a spherical plant. But, plants are made of water, lipids, proteins, lignocellulose. This is how they absorb in the infrared between 1 and 2.5 microns. I want to draw your attention to the blue curve, which is water. So water has an absorption feature here close to 1500 nanometers at about 1450 as its peak. This is a vibrational overtone of water. It's 2 quanta of the stretch and 1 of the bend. So it's also, you're in a solution, so it's a broad peak. But we thought, let's try to image water distribution using ghost imaging. The other thing about 1.5 microns is it's close to a telecom wavelength, where people do a lot of quantum cryptography and key distribution and entangled photon generation anyways. So we knew we could create entangled photons out here. But also the other thing about water is, it's not a trace constituent of anything. It's present at 55 molar in cells. And I'm like, if we cannot see water, then we've got problems. So I thought it made sense to look at water that way. I thought it made sense, again, I'm a physicist. I know more about plant biology than I did a number of years ago. But I don't know much about plants, but I know that water is important. So, we thought, let's start here at 1.5 microns and try to image water in plants. We chose 2 plants of biofuel relevance. One is sorghum, one is camelina. The other thing about these plants is they use different methods of photosynthesis. Sorghum is a grass. It uses what's called C4 photosynthetic mechanisms. Camelina sativa is a leafy plant that uses C3. So these are better in drought. These are better in cold weather. The C number denotes the number of carbon atoms in the first stable intermediate of the Calvin cycle. And so I thought, well, not only are these 2 biofuel plants of relevance to Department of Energy, who's the sponsor of this work, that we could compare and contrast these 2 different photosynthetic mechanisms using a light flux that doesn't really perturb them was sort of the goal of the grant at the beginning. But this is where we started, was trying to image water in living plants at 1.5 microns. So this is what a schematic of our ghost imaging system looks like. We take a laser at 405 nanometers. We use a periodically pulled nonlinear crystal to take that 405 nanometer light and create signal and idler photon pairs at basically 540 nanometers for the signal and 1.5 microns for the idler. And this is the spectrum of our pump laser, the signal photons and the idler photons measured in a spectrometer. But after we generate these signal and idler pairs, we collimate them coming out of the crystal with a parabolic mirror. You've got to use a lot of reflective optics with these 2 wavelengths, which are very dissimilar they're 2 very different wavelengths. So conventional, lenses generally don't work, so you use a lot of parabolics. We then split the light, and then you can start using conventional lenses, but we relay the idler beam through our sample, and then whether he makes it through is detected with a single element detector, a single photon counting avalanche photodiode. This is an indium gallium arsenide SPAD, which can detect light out to 1.5 microns. The signal beam, he goes through free space, and then we record his position on a camera, which is sort of special. We call it NCAM. These things used to be called RULI detectors at Los Alamos, remote ultra low light imaging. But because it's so special, I'm going to spend 1 slide telling you how this camera works. because it's really, I think, what we added to the quantum ghost imaging field was sort of using these single photon imaging cameras to this field, and I think they have a lot of advantages to this field that we were able to hopefully exploit and introduce into this field. So I'm going to tell you about photocathode-based single photon imaging. Again, this is called the nocturnal camera. When we started our work, when I first wrote the proposal 5 years ago or so, you could not buy one of these things. These were made at Los Alamos specifically for sort of national security applications, things like LiDAR. But what these are, these aren't like any camera you're used to. There are no pixels, there are no frames. What this camera does is record individual photon arrivals with about 100 picoseconds accuracy. It's a 1-inch round sensor surface that will tell you where it hit on that 1-inch round sensor surface with about 100 microns accuracy in X and Y. I'm going to tell you how they work. So you take a photon and you convert that into a photoelectron at a photocathode. So once this is an electron, now you can amplify that through a microchannel plate stack. You can take that one electron, turn it into 10 to 7 electrons. That electron cloud then impinges upon an anode array. You have one wire serpentating through, which gives you sensitivity in X. You have another wire serpentating through here. When this cloud hits, it takes a certain amount of time to reach X1. It takes a certain amount of time to reach X2. You measure that time difference, you know where on that wire that cloud of electrons hit, and that's how you get your sensitivity in X location, and you get your sensitivity in Y location from your other cross delay line, part of your XDL cross delay line sensor. And again, they were using this for sort of LiDAR type applications where you shine a pulse laser, look at the time back and get sort of 3D tomography of stuff that is classified and I could not talk about, but I can tell you about the work we're using with this on plants. But what I do think if we did not have this camera available, they would not have funded our grant. This is, I think, a fairly unique thing to the national lab we could apply to this. But now I would say there are commercially available cameras which use this photocathode technology. The one I know of is from Photon Score, a German company. It's called LinCam. And we do have these now also in our lab. And I can tell you about old technology versus new technology. But the key strengths of this is, again, this exceptional temporal resolution. I told you could tell when a single photon arrived with 100 picoseconds accuracy. This is a histogram of arrival times, a correlation between our bucket detector and our NCAM. And so it's got a full width half max of 200 picoseconds, but this includes both the IRF of the SPAD and the camera. So actually the first time, even before we had images and I saw this, that we could get a correlation peak that was 200 picoseconds wide off of a sensor surface that was 1-inch round, I was just blowed me away because a lot of times when you're trying to do fast work and single photon timing, you have very, very small sensor surfaces. So that temporal resolution, again, is very important for beating down noise in terms of making a narrow coincidence window for these two photons. But the other great thing about the sensor surface is it has very low dark counts. It has about 100 counts per second per millimeter squared at room temperature. And that, it doesn't have pixels, but you have an analog uncertainty on your position. But you can sort of, say it's about 4 counts per imaging sensor area. It's about 4 counts per pixel per second. So these are the two key strengths of this for ghost imaging. And then, we built the system, and I was talking about what's like the first thing you image, and me and Doug were talking about imaging beads and how lame that is, and how we always start with it anyways. But this is even probably lamer, is we imaged a razor blade. We just blocked half our beam, and could we see this blocking of half the beam in our microscope? And you could call this lame, but also like it allows you to find your focal plane very easily, but also how sharp this razor blade feature is. If you do a line profile, that tells you what your spatial resolution is. So I have a spatial resolution about 15 microns over a field of view of a few hundred microns. Then you start imaging test targets. So we were doing quantum ghost imaging, so we like to image ghosts from Pac-Man. So this is one of the ghosts we're imaging using quantum ghost imaging in our system at 1.5 microns. It's also obligatory. You do your institutional logo. So this is the Lano logo in the quantum ghost image. But again, we were getting money from the Department of Energy to look at plants. So we thought maybe we should look at plants rather than test targets. But I also want to point out, like other people have tried to use quantum ghost imaging to image biological targets. Well, I would say the targets they were trying to image weren't very challenging. So, Boyd and Paget and others, in terms of this low light paper that came out, which was imaging with a wide field gated ICCD, they were imaging a wasp wing. So a wasp wing, I don't know if you've ever looked at a wasp wing or a fly wing, held it up to the light, but those things are pretty much flippant transparent. They're one cell layer thick. You can basically see right through that thing. So this is not a very challenging target to image through. Another paper which recently was sort of in the plant biology realm was looking at using ghost imaging to look at an onion, a single layer of an onion, a single peel, single peel of that onion. And again, I don't think you've ever looked at a single slice of an onion, but that single peel, it's very thin, it's one cell layer thick, you can basically see through it. So these are not very challenging things to look at with quantum methods in terms of biological samples. And we were interested in looking at plants. And plant leaves and blades of grass, especially on new plants, aren't that thick, but they're hundreds of microns thick. They're also, not transparent. So basically, when we look at a binary test target, something where you get 100% transmission here and 0 here, we're getting, tens of thousands of counts for a given integration period in our correlation peak between our two detectors. You stick a plant in there, it's like sticking an ND2 filter into your system. So if you're in optics, go grab your filter sets, grab your ND2 filters, put them over your eye, walk out into the hall and see how much you can see. But it's basically, it's a much more challenging experiment because you're down 2 orders of magnitude. But because we have such a narrow correlation peak and we can beat down this grass to the side, the other good thing about plants is they don't move or grow very fast. So we can integrate for a while. It's not like we're doing anything great temporally here. We have the luxury to integrate for a while. We were able to do quantum ghost imaging at 1.5 microns of live plants. This is a picture of sorghum on our quantum ghost microscope. It was just sort of growing this way. We taped a blade of that sorghum to a slide here. We're imaging not through the glass and where this thing is taped, but actually just to the side of it. And when we found out, when we started imaging these live plants, we got structures which looked like this. We'd find these sort of rows of circular bright regions. And so these are up to like 5% light transmission on a background of around 1% light transmission at this 1.5 micron wavelength. And so we thought these could be stomata. So stomata are holes in plants. They're holes in plant leaves which are open during the day. So gas can exchange, CO2 can come in and O2 can come out. And they actually close at night so that you don't lose water while you're not doing active photosynthesis. So we thought these were stomata, and also they're these rows. They're around the right size of stomata. They're in rows. Actually, stomata in grasses are known to grow in well-defined rows. But more, so we thought they were stomata, but another key finding was when we were doing our ghost imaging experiments, this is a fairly complicated figure, but basically when we were doing our ghost imaging experiments, we'd find a lot of these bright spots at the beginning of it. And then as time went on, an hour later, we were seeing less and less of them. So it seemed like these were stomata which were closing during the course of our experiment, that the plants thought it's night and they're just closing these off. We actually can see these things disappear, pulse in some light, and have them come back. So it sort of points out that the type of imaging we're doing with this quantum ghost imaging, like I said, we can start looking at what plants are doing with a light flux that doesn't perturb them. It's definitely true because they're going to sleep during these experiments, basically. So we were encouraged by that, and we're trying to get movies. I had that 3D movie of an engine. I do think movies are awesome and cool, but I have no movies of stomata opening and closing to show you right now, but we're working on it. What I will show you is something simpler, which is a movie of binary test targets going through our system, because quantum imaging is now, for these binary test targets and these cameras that record individual photon arrivals, you can now build the ghost image in about a second. So this is a movie of us just translating various binary test targets. I told you like to image ghosts, so we have One variety of ghost here is, if you've ever played Pac-Man, this is when the ghost is chasing you. And then there's the one I showed earlier, which is sort of the scaredy cat ghost when you're chasing it. But he goes through our system here. But now, and here we are using, this data is from a LinCam, not the NCam, but this is from the commercial version of the NCam. And then the next thing which flows through is a cat. Cats are also a very important target in a lot of quantum imaging papers, you'll find out. But then we spend some time on the sedentary cat. But we are now at the point where we can not only do quantum ghost imaging, but we can start to do quantum ghost movies. And we're working to do that in real biological systems, but I have nothing to show you. We're also, in addition to photocathode-based sensors, things like NCAM and LinCam, we've also been collaborating with Eduardo Chabron's group at EPFL in Switzerland on SPAD arrays. So he's really been leading SPAD array development for a number of years. We've been working with him on a SPAD array called Piccolo that is a 32 by 32 element SPAD array. And we were comparing and contrasting in this paper what you could do with these photocathode-based imagers, things like LinCam and NCAM versus SPAD arrays. SPAD arrays, your temporal resolution isn't as good, and your dark counts are higher. But what's great about SPAD arrays is it's like each individual SPAD in this 32 by 32 element array, it's its own detector. So you have a lot more parallelization. You can detect more photons per second. This thing, LinCam, these photocathode-based LinCam and NCAM, they saturate. You detect one photon over the entire sensor surface, and that thing's brain dead for a microsecond. So you can only do this at about a megahertz detection rate. So there's only so fast you can image with this. This can image much faster. But I would say because you have lower noise and better temporal precision on these photocathode-based sensors, they're much more better for precise low transmission measurements, whereas this is better for fast imaging and potentially faster dynamics. So in addition to when we started this project and building up our ghost imaging capabilities, we were like, well, we can image in the infrared, and it's like, what can we learn about plants with infrared spectroscopy, and where should we be looking? I mean, I told you my rationale for looking at 1.5 microns, but are there other wavelengths which are more important that would tell us more about plant health or plant resiliency? So we just started doing a lot of conventional measurements in a FTIR microscope with sorghum and camelina, the two plants we were interested in, and looking at changes in their IR absorption spectra as a function of various environmental stressors, growing controls, cold, drought, salt, stress, salt, shock. And there are various, you can assign functional groups or groups of functional groups to a lot of these bands. And we did find that you could use FTIR and infrared imaging to infer plant health and classify, is this plant healthy or not? You've got full spectral data. You can do all sorts of, principal component analysis and clustering type stuff to classify whether a plant's healthy or not. But, so, we hope to get this published at some point. We've all been down that road before, but you can read about this if you want on the bioRxiv server. But what I want to point out is all of these measurements we were doing with a commercial FTIR, you're going, a commercial FTIR, the one we have, this Bruker, goes from about 500 wave numbers to about 4,000 wave numbers. I've lived all my life in nanometer space, so it goes from 10 microns down to below 3 microns. And these features that I've been talking about in terms of, say, OH stretch, which would tell you about water and polysaccharides, it's at about 2.8 microns, 3,500 wave numbers. It just goes further into the infrared from there. So the problem with ghost imaging at these wavelengths, you can generate entangled photons out here, but for ghost imaging, you still need to detect that infrared photon, whether you saw it or not on a bucket detector. And so that bucket detector needs to be single photon counting. And single photon counting out here at this wavelength is very challenging because this photon does not have much energy. There's superconducting nanowire sensors, which are getting further out into the IR now. It's where these are nanowires which are superconducting. You hit it with a photon, it raises the heat enough to change it from superconducting to normal conducting, and you measure that. And so you can do single photon counting with nanowires, but no commercial things exist out at this low a wavelength. So there are no good single photon detectors out here. So we were interested in developing and are working towards developing a different quantum imaging technique that was developed by Anton Zeilinger in his lab and Gabriele Limos and Sven Reimelo and a few others about 10 years ago, called quantum imaging with undetected photons, or QUIP. And what's good about QUIP, I'll explain how this works right now, but here I'll just say it's similar in that you're creating entangled photon pairs in a nonlinear optical crystal. But in QUIP, and you're using the Idler photon to probe your object of interest, you're forming an image with the signal photon on a camera or an imaging sensing detector. But what's interesting and not obvious how this works is actually the Idler detector in this scenario, you don't even need to detect it. It goes off into free space and lives its own happy life. And so what's good about this technique as opposed to ghost imaging is you don't need to detect this Idler photon. So you can potentially get this information-rich fingerprint region in an imaging experiment with this technique. And so I don't know how much time I have. Well, I guess now I do because I looked at my watch. But I'll tell you, understanding ghost imaging is way easier than understanding how QUIP works. And so, I'll just say, I'll show you a little bit how this works, and I'll try to explain how it works. But it's I think much harder to conceptualize how it works than ghost imaging. But you take a laser and you have a nonlinear optical crystal here where you're creating signal and Idler photon pairs. Your Idler photon reflects off a dichroic, goes through your object of interest, reflects off another dichroic, and go through a second nonlinear optical crystal and then out into free space. So this is your Idler path here. Your signal photons in this thing come out of your nonlinear crystal. They can come off a dichroic, come through here. In this experiment from 2014, their signal photons were at 810 nanometers. Their Idler were at 1550. And what you find out is that you can form an image with this signal beam of this object, even though, again, the signal beam never goes through or samples your object, only the Idler beam does. And I think understanding how this works, again, I said was difficult. I'm going to try to explain it with another experiment, which I think is very difficult to understand how this works, but at least we've all seen it before. And that's the Young's double slit experiment. And so, we all know that from high school physics, light behaves as a wave. If you have a point source impinging on two slits, you form a far field interference pattern. These things interfere like it's a wave phenomenon. But we also now have detectors which can detect single photons and single arrival events. If you redo this experiment with those types of detectors and you start building up single photon positions in the far field, after a few photons hit, you'll see this more and more. Ultimately, you'll rebuild this interference fringe pattern. And so the question is, you're sending photons through one at a time. How could they interfere to give you this fringe pattern if you're only sending one through at a time? And that's the thing we don't really understand very well. It's like the photon senses both paths, or you can write out both paths. It's almost like it senses both paths, it interferes with itself. Sometimes it's called photon self-interference. You could think of this as a sum over histories approach to quantum mechanics if you're Feynman, that it's like it sniffs it all out and you have a probability here at the end. We all learn how to sum, wave functions, which, we don't know where it came from. So it could have come from each, and we sort of like take this complex number, the complex conjugate, square it to get some probability distribution, and you get this in the end. But I think my little Newtonian brain, it has a hard time understanding this experiment, that you could get self-interference with a photon. But I guess I was talking earlier, it's like it behaves as a wave until you detect it, and then it behaves as a particle. If you're into the Marvel universe, there's one photon where this went through this slit, there's another universe where it went through that slit, and this is the interference pattern from those two different universes colliding. But in any event, I don't think too many people understand this. It really defies our classical brain. But I spent a lot of time on that because the two nonlinear optical crystals in QUIP are a lot like the two slits in Young experiments. You could have an entangled photon pair that's generated here, or an entangled photon pair that's generated here. Because you don't know which one of these two it came from, they're indistinguishable. It acts as if it came from both. And actually, when you go through the math of the idler photon being absorbed here or not, this idler photon being absorbed here or not messes with the interference pattern. It is imprinted in the interference pattern of the signal photon. So again, and it really is interference. It's like single photon self-interference. It's more complicated because you have two photons here and two photons here than the Young's experiment, but you can get, a certain distance, you can get constructive interference. You can get destructive interference at a different distance. So this is really single photon interferometry. And so if you get these interference patterns at different distances away from your system, either constructively or destructively, it might remind you of how an FTIR works in terms of scanning an end mirror and getting an interferogram. And then an FTIR, you take the Fourier transform of that to get your absorption. But there is a group in Japan, Takeuchi's lab in Japan, has been doing a lot of work that I think builds off of QUIP. Instead of imaging, they're doing single point measurements, but they're calling it quantum FTIR. But they are scanning a mirror to build an interferogram and actually taking FTIR spectra with quantum entangled light. It's actually a fairly small footprint instrument. What we wanted to do was to take what Takeuchi had been doing with sort of broadband spectral information with quantum entangled light and combine that with the imaging information of QUIP. We're very interested in trying to combine these two techniques so we can do sort of hyperspectral imaging in the infrared. And so that's what we got money in our renewal grant to try to do that. So I can't show you results, but I'll show you where we stand on the path to results towards that. Here. So I haven't talked too much about how you take one photon and split it into two. I told you it's this process called spontaneous parametric down conversion. You use nonlinear optical crystals to do that. To create two photons from one, you have to match, those two photons have to sum in energy to the photon of the original. They also have to sum in momentum, which we did talk about a little bit. But how do you get the momentum of these other photons to be the same as your original one in an optical crystal? You basically need to match the index of refraction of your pump, signal, and idler. And so if your signal and idler are the same energy and they're degenerate, oftentimes you can match the index of refraction in a birefringent crystal, something that has an index of refraction for one axis, which is something, and a different value for this. And a lot of times you can angle tune this to get different wavelengths. That only works if your signal and idler are very close in energy. If your signal and idler are very non-degenerate, like we want to do for a lot of these imaging applications, then you have to start using man-made artificial materials where you modulate the index of refraction using periodically polling, or you modulate the nonlinear optical susceptibility of your system. But I'll just say, if you use this mismatch in delta K of your material to make up for the mismatch of delta K of your photons. But what you have, if you have an even polling spacing, you can create sort of a narrow, nice, well-defined energy in your signal and idler spectrum. What Takeuchi did was, instead of a single crystal with well-defined, he chirped it, so you had different periods where you have some at this spacing, others at this spacing, sort of a gradient over a 1 centimeter long crystal. So you can generate different wavelengths at different sections of this crystal, and they were actually able to create a fairly broad band entangled photon source for their quantum FTIR that went between 2 and 5 microns, 2,000 to 5,000 nanometers, or between 5,000 and 2,000 reciprocal centimeters. I'd say the other thing I like about this figure is I'm constantly reaching for my calculator to convert wave numbers to nanometers. And this is just flipped. It's actually 2,000 to 5,000. It's 2,000 to 5,000. It's just backwards. So in any event, so we saw this, and again, we wanted to apply this to imaging. But the problem with this 1 centimeter long crystal, this chirped polling period structure, is it's not compatible with imaging. This centimeter long crystal in the way you image, I talked about momentum conservation, but also a lot of times you're relaying the image of the crystal face onto your camera. And so you don't really know in this long a crystal where your photons were generated. So our crystal is only like 1 millimeter long for the ghost imaging experiments, and thinner crystals give you better spatial resolution. So, this chirped approach, you can't use a long crystal, but we thought we could use shorter crystals and a fan-out approach. And so you can also not just pull the whole material, you can actually pull it in what's called a fan-out geometry. And then you can tune the polling spacing or period and the wavelengths which are generated by translating the crystal side to side of a fixed laser beam or translating the laser beam fixed side to side. You go through different polling periods and produce different colors of light in your signal and idler photon pairs. And so, you could move the crystal side to side, but it's always faster to fly a laser beam around with a galvo mirror than it is to physically move a crystal. So to do this fast, Duncan Ryan, who's on this project, and I'll mention at the end, came up with a way that you could use a scan and descan approach with 1 galvo and 2 sets of different fan-out crystals, one where you're scanning it back and forth in a separate galvo mirror, which descans it. So if you ever used a confocal microscope, you have 2 sets of galvos, one to fly it around in X and the other in Y, but you actually have a stationary pinhole for the resulting fluorescence, because the same scanning and flying the beam around also descans your fluorescence. So this is sort of a scan, descan approach. You need to duplicate this for 4 crystals for the coverage we wanted. And we did just get a provisional patent application on this scan and descan approach. I'll just show you what it looks like in the lab. This is, with the laser on and everything pretty, but these are sort of the 4 fan-out crystals we have, PPMG, SLT. This is the entangled photon spectrum from a single crystal. So this dotted line is what the signal photons look like at a single fixed galvo position. When we sweep the beam, it's this solid green line for that 1 crystal. This is what the idler correspondingly looks like. And this is a log plot. That was the other thing, is Takeuchi's thing looked really nice and flat over this whole wavelength range. And then we realized, hey, he also took the log of it. But you live and learn. But in any event, we have a fairly nice flat distribution, from this 1 crystal from 3 microns down to about 2.5 microns. But this is just one of the 4 crystals. These are the spectra in the signal for the 4 at 2 different galvo positions. And then when you sweep this, we cover, basically in the signal's not important. The idler's the one that's important because that's where we want to do our sensing. We can cover actually from 2 to 7 microns. So we have a broadband entangled photon source that we're trying to now do hyperspectral imaging with in QUIP. And I don't have any of those results to show you because they don't exist. But that's why we're, this is science. And I didn't want to just talk about stuff I've already done. So where we're headed with this is, we want to do QUIP in the fingerprint region of the IR spectrum because it's much more information rich, not only for plants, but for lots of things. They call it the fingerprint region for a reason. But we also still, because we have a quantum ghost imaging microscope, and we want to, build up. We started at water, and I told you why we started at water, but these other dashed lines are also where we would like to go with our imaging. We'd also, I think our next wavelength will be at 1200 nanometers for the idler photon. And what's great about that is this is where lipids absorb more and water's down by an order of magnitude. So we could get a water image, we can get a lipid image, and hopefully, start to build up what's all in the sample at these discrete wavelengths. Okay, so the take-home message, what I want you to remember from this is that the power, I think, of these quantum methods, whether it's QUIP or QGI, is that you can probe at 1 wavelength and you do your detection at another. And that really allows you to start to be doing imaging in the visible, but probing in the infrared. And we don't have good infrared, they're infrared night cameras and stuff like that, but they don't go that far out into the infrared. Mercad telluride is a terrible material in terms of its dark counts and need to be cryogenically cooled. This is, I think, very powerful. The other powerful feature of ghost imaging in particular is that because we're detecting single photons and doing this correlation, we can do this at a very low light flux. So, we got money to look at plants where this also makes sense. But there are other biological systems where this also would make sense. Anything with a photoreceptor, the human eye, a cow eye, anything where you shine light on it and its physical state changes and you want to see what it looks like in a resting state, that makes sense. The other thing, I'm in a materials physics and applications department, so people are all like, well, what can you do with materials? You're always looking at cells. There are materials which are light responsive polymers, photoresists, solar cells. There are also materials where I think low light imaging can make sense. So that's the take home message. I want to acknowledge the funding sources. So Department of Energy, BER, BSSD, BCIS. I can tell you what all these acronyms mean, but biological environmental research, they really were the driver and the primary source of the funding for everything I've talked about. I also have some money to do quantum imaging and quantum sensing from DTRA and from LDRD, which is an internal laboratory fund. I did want to acknowledge Duncan Ryan. He built up our ghost imaging microscope. He also is the whole driver of this broadband entangled photon generation. So his work here is pretty much everywhere. Katie Seitz was a plant biologist, a postdoc on this project, who's been with us from the beginning, and I'm so glad she wasn't frightened by all our lasers and optics. And she's been growing all the plants we've been doing all of the conventional biochemical type measurements, all the IR measurements, and also we've trained her up to use this ghost imaging scope. But she's also been very important to this project. I'll say I know nothing about plants, and that's why I have Dave Hansen. Actually, I think I did a mental blank. I put ASU here. He's from UNM, and when I don't know why I did that, but Dave Hansen's from UNM. I think when I was putting this slide together, at least I pulled the right thing from here. I was moving his names around, but this is back I was visiting here. I said he was from ASU. He's from UNM, but he's also been a great collaborator. And then the people at EPFL provide us with the SPADs. And then that's my last slide. So I will take questions.